This website aims to answer the question, how do anti-anginal drugs switch metabolism in the heart from fatty acid to glucose oxidation to improve symptoms that arise from myocardial ischemia? To begin, we will first talk about the differences in fatty acid and glucose metabolism in the heart. When a person consumes a meal, food is broken down into free glucose and free fatty acids, both of which make their way into the bloodstream. These molecules can be oxidized by different metabolic processes to yield usable energy for the body in the form of ATP. Now, we will briefly discuss the fundamental metabolic pathways that occur in cardiac cells. Specifically, ATP can be synthesized by glycolysis, glucose oxidation, and fatty acid metabolism. Glycolysis is an anaerobic process. Every glucose that enters this pathway yields 2 pyruvate, 2 NADH, and 2 ATP molecules via substrate-level phosphorylation. In glucose oxidation, pyruvate produced from glycolysis enters the mitochondrial matrix as acetyl-CoA, which then enters the TCA cycle and generates NADH and FADH2 molecules. The reducing equivalents produced up to this point enter the ETC to yield 38 ATP molecules via oxidative phosphorylation. Fatty acid metabolism is another aerobic process. Within the mitochondrial matrix, fatty acids undergo several rounds of beta oxidation to yield a large amount of ATP molecules. This is the preferred source of energy in cardiac cells. So this prompts the question, why does the heart prefer to oxidize free fatty acids and not glucose? What's the difference? Well, we can compare glucose with the fatty acid hexanoic acid because both have a 6-carbon backbone. Oxidation of glucose and hexanoic acid yields 38 and 44 ATP molecules respectively. Fatty acid oxidation is preferred in cardiac cells because it is more energy efficient since it produces more moles of ATP per carbon oxidized. In contrast, glucose oxidation is a more oxygen efficient method because this process produces more moles of ATP per mole of oxygen consumed. Myocardial ischemia is defined as an inadequate supply of oxygenated blood to the heart muscle. This results in an increase in fatty acid concentration, decrease in ATP synthesis, and increased acidity from lactate accumulation. The combined effects produce symptoms of angina. Angina is defined as chest pain or discomfort that occurs in the heart. It would be beneficial for humans to use glucose, a more oxygen-efficient fuel, as the primary source of energy during ischemia. However, it is important to note that humans cannot voluntarily switch between oxidation of glucose and fatty acids. Anti-anginal medication is a promising strategy that can be utilized to treat those suffering from angina due to ischemia and is believed to be achieved by inhibiting beta oxidation and increasing oxygen-efficient glucose oxidation. And this prompts the question, how do anti-anginal drugs switch metabolism in the heart from fatty acid to glucose oxidation to improve symptoms that arise from myocardial ischemia? Thank you for watching our video. Please check out the rest of our website for more information about the metabolic pathways of specific anti-anginal drugs.